2022, also called the RGB Festival this year for Red, Blue, Red, Green, and Blue. Sorry. So I'm Thibaut. I'm the founder and the director of Paint of Science Norway. And if you are the first time you come, you might wonder what is Paint of Science. So it's a worldwide movement born in UK 10 years ago. Oh, it works, perfect. And the goal is to bring scientists into bars and to engage with them. So in a better and relaxed atmosphere. And so it's 30 countries nowadays. On this festival, there's 25 countries having every night.
That's why.
So now we get it. Just unplug it.
Unplug it.
Yeah, unplug. Is that better? Thanks. So as I'm sure, so yeah. So this festival is 25 countries and is organized by volunteers for you. And this is Barney, our mascot, which is actually here handmade by one of the volunteers. And in Norway, we are present all over we have an exponential growth until COVID, of course. But we are back. In Norway, we are 2.5 years old, and this is the third edition, but the two first were online. So this is the first time we host in person, and we are very happy. So you can find us in 5 cities at the moment in Norway. So I'll see Tromsø, Trondheim, Bergen, Stavanger, and Oslo. I'll say the back. And you can find as well, we have the website, the main source of information, the paintofscience.no, and we try to be present in all your social media. So you can find basically us everywhere. Be aware that tonight is streamed as well on YouTube, on Facebook and LinkedIn. So the webcam is that direction. So we won't see you unless you pass that way. And now I let the stage to Lavinia, which is our Oslo City Coordinator. Thanks.
Thank you. Thank you, Thibaut. Thank you to all of you for being here tonight. So I'm here basically to say, maybe, yes, to say thank you to a lot of people. First of all, to all the volunteers that worked very hard for the organization of this festival. As Thibaut said, Paint of Science is created entirely by volunteers. So I'm really grateful to all of them because they did a great job. They worked a lot. And I want to thank Dane, who worked together with Chan, that is not here tonight, and Judita to find all the speakers, and is also working on the social media together with Agnieszka, which is our social media manager in Oslo, but also the coordinator at the national level. Praveen is the IT guy, so is the IT coordinator at the national level, is also, of course, taking care of everything concerning the technical issues here in Oslo, together with Thibaut. And a big thank also to all the other guys that you can see here in the slide. So Lourdes, Lourdes, that works a lot together with Judita for this edition to create all the quiz. We will have a quiz later. And she also worked a lot with Praveen and Maren, that is not here now, to find the locations. It was a very hard work, so thank you very much. And Maren, as I said, is another volunteer who I must thank a lot, because she's the person who entirely translated the website in Norwegian for us, because she's the only Norwegian volunteer in the team. So thanks a lot. So thanks to all the volunteers, thanks to all the guys, but of course thanks to our speakers, because the work that they are doing here tonight is particularly complex, particularly hard. We know that we are asking you to work a lot to prepare this kind of presentation, so we are really grateful for this, for your presence here. Tonight we will also have 2 presentations given by two of our volunteers, so from Lourdes and Agnieszka, and the third speaker for tonight is Elisabeth, and I really want to thank her for this contribution. And again, the last acknowledgement for tonight goes to our sponsors, because we couldn't do anything of this without our sponsors. Simula is supporting Pint of Science Norway since the first year, so since 2020. This is the third year, together with Comites, that is also supporting since the beginning of Pint of Science Norway. And this year we have a new sponsor, that is NMBU. So thanks a lot. Before leaving the floor, I'm sorry. Yes, before leaving the floor to Praveen, that will also present the quiz for tonight, I want to ask you to send us your feedback, because they're important for us. So let us know what you think about this festival, what you think about tonight. It's really important to improve every year and try to make it better next year. And of course, if anyone is interested in science communication and wants to help and wants to be part of the team, please join, because there's always work to do. The team is pretty big, but as we said, we're all volunteers. So if we can share the work, it's even better, because the work is really a lot. So we invite everyone to be part of this journey, and we hope it could be something funny for everyone. So now, as I said, we have a quiz. Thank you, Praveen. Praveen will share the quiz tonight. Yes. Please join, because there's a prize for the winner. Yes. Yes, just a second. Just a second. We need to change the presentation.
Oh, yeah.
Okay, then I will go there.
What?
Yeah.
No, why is it only the board?
Okay. Yeah, just give me a few minutes. We are trying to sort out the technical problem. Okay.
Yeah. What is question 1 to?
I don't know what you're trying to say.
Now we speak.
Yeah. Yeah, I'm good.
Okay.
Okay. Welcome to the event, everyone. So we are going to have a quiz of 9 questions. It's mostly on the topics from the today's discussion. So it's more like to make sure that you are prepped for the talk. So the quiz will just give you the introduction, and then like the presenters will give you a good talk. And yeah, I think yeah. Okay, now it works. Okay, I think I will wait for like few more 1 minute to make sure that everybody has joined. Okay. Okay. Right.
No, it's me.
Hmm.
I'm doing.
Okay, so I hope everybody has joined the quiz. Let us start. Okay, okay. Uh, yeah, it's working. So the first question is, who are the number one countries catching Atlantic cod? So we have three options. First one is Norway and Portugal. Second one is Iceland and Norway, and third one is Brazil and Japan. And time's up. Yeah, it's Norway and Iceland. So yeah, this fish it grows in it more or less it's very popular in like one of this Norwegian sea. That's what I have read, and probably Lud will tell you more about it. Let's see the winners. Ah, okay, SMG. That was a good one. Okay. The second question, I have to enter. Yeah. What is that word? I don't know. And yeah, the three options are eating soap, being afraid of soap, and allergic to soap. Ha! Eating soap. Let's. Okay, we have new winner now, and people are close. Let's see. Okay, the third question. What is N2O? Yeah, let's see how the answer comes up. Yeah, it seems like most of you are like very familiar with all the scientific term words. Good luck. And yeah, we have the same winner dominating the board. Okay, according to anecdotal sources from the medieval ages, where could you find the largest schools of Atlantic cod ever known? So you need to know what is school here. It's kind of like a way the fishes socialize together. Like they will move from one direction to another direction. Yeah. So yeah, if you go through Wikipedia, like in that particular place, there has been a big fishing industry from the last 500 years. Okay, the new winner, Mr. G. What is soap made out of? Yeah, that seems like a complex question. Let's see. Yeah, we have three options: plant seeds, oil, water, sodium, water, dirt, sodium. It seems like water is very common here, but you have to guess the other two. And yeah, most of you have guessed right. Right? Good. Ha, Mr. G, you have lost it, and now it's Azan. How would the world look without greenhouse gas? Colder, warmer, the same? Ah, yeah, that's a good question. I don't know. Colder. Let's. Ah, yeah, there is a very close race for the top position. So, approximately, when did people started consuming fish? 2,000 years ago, 40,000 years ago, and 10,000 years ago. 40,000. That's a very large number. Ah, the board has changed again. Why is soap harmful to environment? Yeah, you have four options. Let's see. Ah, all the above. Oh, okay. It's a close call, and we have reached the last question. How much does N2O heats up the earth compared to CO2? 300 times, 1000 times, two times. Uh, okay. And the winner is. Okay, you didn't see the answer. Fivers. Congratulations. Okay, the answer to the last question that's missed, it's 300 times actually. Yeah. And the winner, can you come here? Like, we have a gift for you. And we are working on it. Okay, that is. Okay, it's failure. Yeah, yeah.
Thank you. And yeah. And the other one is
It was very low.
Are we ready? No?
Thank you. I'm sorry.
Can I use? Hi, everybody. So my name is Danae, and I will be the chair for this night. Today is our third day of the Pimp of Science, and maybe you know that this year we are having a festival with the thematic of RGB, the spectrum of sustainability, and of course we already have the red day and the green one, and today we have the blue day. This day is about topics related to whatever you think about blue. For example, water, sky, and other things that our speakers will talk about. Our first speaker is already here. Her name is Elisabeth, and I will read you some of her story. So, during her teaching education, Elisabeth found her interested with geology. Leading her to a master project on how bacteria influence the breakdown of minerals. This introduced her to the world of microbiology with a particular interest in microbial nitrogen cycle. After 2 years working as a lab technician, she decided to take on a PhD studying the use of bacteria to mitigate laughing gas emissions from agriculture. She's looking forward to talk with you about this research tonight. So let's hear about her topic.
Okay, thank you very much for the kind introduction. I'm, as you said, really looking forward to talk with you tonight about how to fight climate change with N2O-reducing bacteria. So this is the outline of my talk. I will briefly talk about climate change and greenhouse gases. Then we will have a closer look at laughing gas, or N2O. Then we will touch upon fertilization, food production, and soil bacteria that produce N2O. Then we will take a look at how soil bacteria might be a solution to the problem. And I'll show you some promising results. From field experiments. Computer? Okay. When I say climate change, you might think of extreme weather like drought, floods, polar bears on melting ice flakes. But you might also think about carbon dioxide from driving cars, methane from burping cows. But have you heard about laughing gas? I guess you've all heard about it as a sedative used in hospitals, but it is also a very powerful greenhouse gas. Let's start with some facts. When N2O is released into the atmosphere, it stays there for a very, very long time. This means that the N2O we release today will be a problem for us within the next 100 years. And when it's up there in the atmosphere, it breaks down the ozone. And as you might know, the ozone layer is like the Earth's sunglasses. It protects us from the sun. But what you maybe didn't know, or what you might have learned from the quiz before, it heats up the Earth about 300 times more powerful than CO2. And N2O, like as we call it, or laughing gas, which is the common name, is currently responsible for about 7% of the global warming. And while emissions of CO2 are going down, we see a downward trend, the atmospheric concentrations of laughing gas is the opposite. They're increasing steadily, and they have been doing so ever since we started putting industrial fertilizers into our fields. So this is a good reason to look into where N2O comes from. And about half of N2O in the atmosphere comes from natural sources like oceans, forests, soils, etc. But a bit less than half of it is human caused by our activities. I'm sorry. Okay. And of the human caused activities, about half of it comes from agriculture or food production. So you see there are different sources, but the main driver of N2O emissions from human activities comes from agriculture. So to produce enough food for a growing population, farmers apply fertilizers to increase plant growth. And we can use industrially produced fertilizers, or we can use, this thing is not on my team today. Organic fertilizers, like sewage sludge, or cow manure, or straw, or anything else. In our project, we are using organic waste materials as fertilizers, like sewage sludge or cow manure, because they are already a part of a circular system where they are collected and then they are put back into the soil to add nutrients for plant growth. But it's not only the plants that take the nutrients we apply to the soil, like nitrogen. Nitrogen is what I'm most concerned about. Soil bacteria, okay, we'll have to do a couple in a row, I think. Soil bacteria are also using nitrogen as their food. And when soil bacteria use nitrogen as their food, N2O is produced as a byproduct of this process. And when the N2O is created under the soil, it leaks out to the atmosphere. There are some measures we can take to reduce N2O release from soil. We can prevent soils from being acidic by liming them. We can fertilize precisely and not over-fertilize. But that's not good enough, because as long as we continue to add extra nitrogen into the soil, we will have excess nitrogen being produced as N2O, and it leaks out to the atmosphere. So we need new solutions to get rid of N2O. And luckily for us, some bacteria use nitrogen and produce N2O, and others take this N2O and reduce it. They transform it into harmless N2 gas. N2 gas is what makes up most of the air that we breathe now, completely harmless. So these are the heroes of my story, the N2O reducing bacteria. And if we could increase the amount of N2O-reducing bacteria in soil, that would reduce the amount of N2O we release. Okay, thanks. Okay, so there are a few challenges to just increasing the amount of N2O-reducing bacteria in the soil, because it's almost impossible to just do something with the soil and expect there to be more N2O-reducing bacteria. And also, we can't just take any N2O-reducing bacteria that we have in the lab, put it onto soil, and expect it to survive and function the way we want. So, our group, with some of my friends here, Kjell and Silas, developed a novel method. I'm sorry, I skipped a part. I need to tell you why this is a problem. It's a problem because soil is a complex material, and resources like food and oxygen in soil are limited, and they are constantly varying. And there are already billions of bacteria in the soil, so the competition for food is hard. To overcome these challenges, my friends have developed a new method to carefully pick out N2O-reducing bacteria that we can use to apply to soil. And that's the new method. Let me tell you about the new method.
It's just me, probably.
I'm sorry.
No, it's fine.
It just confuses me a bit. Okay, so what's new about this method? The new thing is that we create an environment with a mixture of organic material and soil, and we give it N2O as an atmosphere. And in this environment, N2O-reducing bacteria are getting an advantage in the competition against all the other bacteria. So they can grow and multiply much more than the others. And thank you. Maybe I'll just give you a nod and then you can click. Okay, thank you.
Yeah, you can just click me, you know.
Good. And then we can isolate them. And since we have used a mix of organic material and soil, we have then isolated bacteria that can grow in both organic material and soil. And then, of course, we have to do a lot of tests to see that we got the right bacteria, that it actually survives in organic material, like sewage sludge, cow manure, etc. But the idea is that if we can grow N2O-reducing bacteria in large numbers in sewage sludge or cow manure or any other organic material, and then spread it to the field, I like to think of it as a blanket, kind of covering the field with N2O-reducing bacteria, preventing N2O release to the atmosphere. And these bacteria have then the advantage of we picked them, we found them, we know that they survive in both materials, and we also apply them to the field in their favorite material, so they have a head start. So, but even if it works in the lab, how does it work in real life? We do a lot of stuff in the lab, and it doesn't always turn out to work in the real life. But last year, we did a field experiment in the summer, and I would like to explain a bit about the setup first. Here we have a set of pots containing different types of soil, because agricultural soil in Norway can be anything from high pH to low pH, sandy rich, clay rich, different textures, etc. So we tried out different types of soil, and then we grew our N2O-reducing bacteria in large amounts in the organic fertilizer, in this case, sewage sludge. We applied it to the soil as a fertilizer. And then we could use our one-of-a-kind field robot, developed by another friend over here, to measure N2O released from the soil. So these are gas chambers, and it runs automatically over the field, or in this case, the pots, measures N2O, and we can compare it to our control, which of course does not contain N2O reducing bacteria. And we got promising results. Compared to our control, applying fertilizer with N2O reducing bacteria means a 40 to 95% reduction of N2O emissions. And what surprised us more, because we kind of hoped for this, but what surprised us more was a very long-lasting effect. We left the buckets outside for 90 days, just applied some fertilizer without N2O reducing bacteria in the meantime. And even after 90 days, we saw a 50% reduction in N2O emissions. So we will do one test now because this was in the autumn, and we have one more test to go to see how long it lasts. So what's next? Upscaling. Because if this is going to be a success, of course, we can't keep on doing 15 liters batches in the lab. We have to upscale to hundreds of liters for farmers to be able to spread it. And we want to test different types of organic waste materials. This is the good thing, of course. We use materials that are already collected, right? The pipeline is already there. Cow manure, sewage sludge, straw, it's already collected. And then we can inoculate it, we can let the bacteria grow, and we can put it back on the field, which would have happened anyway. We just kind of sneak in this pipeline, add our bacteria, and make a difference. And we would like to find new N2O reducing bacteria for these different types of materials. I'm really looking forward to working on this further with the rest of these amazing people. Thank you.
Thank you for your talk. It was really nice. Is there anyone who has questions? Okay, that was fast.
Yeah. I think I have few. So first of all, the robot that you have, I understand that it's going from pot to pot. You are driving it, like you are doing like it's going sequence. But let's say that I don't know that, okay, it's 6 in the morning, so right now we are measuring, because I understand that it's only one, and it has to go from one to the other. So then like you have like certain point of the time of the day that it has to be measured. Or it's just random?
So we have to take the robot out, but then it's programmed. When we put it in the starting position, it's programmed to measure all the points we wanted to measure. So it can also run in a field. We have a lot of field experiments with it too. But then, we did it to the same time every day so that we could follow the emissions.
Yeah. So the other question about the robot, you have it as a, it's measuring N2O in the real time, or you just capture it and then you send it to the lab and do, I don't know, GCMS or something?
Yeah, we measure it and then we take it in for analysis.
Yeah. So it's not
But you can watch the screen of the instrument to see the N2O emissions as it runs. But it's so much data, so you have to. But you can stand there and watch it and say, oh, today it's really high measurements because it's been heavy rain, or we just applied fertilizer, or so you can use it to see. Now the peak after fertilization is over, for instance, you can say.
Okay, so the last one from me. The bacteria that you're using, it's just one bacteria, or it's just like took it, you analyze it, you know what bacteria are exactly there. So you use it like a group of bacteria, not only one.
For now, we have one, and that's what we're going to work on with. We have several types, but we have one that's like the best. So that's the one we've worked most on for now. But Silrune, who's there, isolated a lot of different N2O-reducing bacteria. But we have one called Cloacibacter, which is a very efficient and good bacteria to use. So, but our aim is to find different, to have more for other organic waste materials too.
That's it.
So you showed the isolated growth of NO2 in the environment in the beginning. And I wanted to know what's the absolute impact of NO2 in the environment compared to the CO2.
So, like how much of the global warming it accounts for? Yes, it accounts for about 7%. So it's smaller in amounts than CO2 and methane. It's the third most important greenhouse gas. But the most worrying part is that it's continuously increasing while the others are going down. It doesn't seem like we have any good solutions to make this decrease. So that's why we worry about it.
No, this.
Okay.
So thank you for the talk. I think it's very exciting. It seems like it's like you found a solution to moving forward. But I wonder, like, if you could maybe give a guess of, like, do you think it will take a long time before this becomes practice? And then the other question is, maybe could you explain, like, I'm just curious to know, like, how do you isolate that? I can imagine it's super hard because there are many.
You
can keep the question to the.
You want to help me out here? I think you know more about upscaling than I do.
There we have when it comes to more kids of the law, that's a big area. But we have we're collaborating now. We have some we are working together with the science association. So it's all about producers and users and regulatory and more positive, but it's 20 hours.
Like, why is it not allowed? Why is it not allowed?
Whether it's a definition of people, as soon as we do something with the medical answer, it doesn't fit into the process. Whether it's also open up to make the box like a new box for this. Secondly, when it comes to isolation, it's basically very hard work.
Done by Sid.
And of course, some thoughts as well when it comes to the selection machine, which is select for the right type. But mostly it's hard work and
I was wondering, when it comes to the production, does it affect having these bacteria? Does it affect the production rate, or it keeps the same? So it doesn't affect what you produce and have you have you done research on that?
No, not yet. We will have to do that. I do not think it will affect it, since it only takes the anthrol that has been produced. It doesn't take the ammonia or the nitrate from the soil that the plants can use. We did grow some grass in the pots last year, but we were better at growing weeds than grass, so that has yet to be tested. But I assume it will be affected much.
You mentioned that it takes about 20 years for that nitrous oxide that's quickly released into the atmosphere to quite less purity. How does it become? Is it a chemical reaction in ozone or something else?
Yes, it's chemical reactions in the atmosphere. Yes.
So I actually have one question. So this bacteria you have tested in Norway that has like really specific conditions. Let's say it's cold. So can this bacteria grow somewhere else where it's more warm?
Okay, so since it's about temperature.
Yes, mainly.
Yeah, I think they can. You can also do these type of. You can use this method for different kinds of soils or in different environments. So that's the good thing about this method that Kjell and Silas has developed. And Christina has tried to use it on African soils, which was a bit more difficult, I think. It's very weathered soils. And for this specific bacteria, we know that it grows, when we grow it, it grows very well when it's warm, or it likes 20 plus degrees. You can.
And then they have another question. It's more like in an ecological way. So you are putting a bacteria outside, and it's a bacteria. I don't know if it's actually always there, but you are putting it in big quantities. What will happen with the rest of the population of bacteria that are not this species? Will they die, and then, or I don't know, whatever they are supposed to do, these other bacteria, what will happen?
That's a very interesting question, and we can't answer that clearly right now. We were very surprised, as I said, that it actually had an effect after 90 days. We thought it would fade out much quicker, because that's what we've seen on applying bacteria to soil before from other studies. So that was very surprising, and as I said, we will do one test now soon when we have the time to check if there's still an effect. But we don't really know what it does to the soil environment yet.
I think that there's another.
Skal jeg si noe om sopp? Skal jeg si noe om sopp, eller skal jeg ikke si noe om sopp? Nei. Nei.
Yes. I think that there are no more questions, right? So let's give a big clap to Elisabeth. Thank you very much for your talk. And we're going to take a break of about 10 minutes. So just go and grab more beer or go to the toilet or whatever you need to do. See you in 10 minutes. are going to continue with the next speaker. Okay, so
For the camera.
sorry guys. Yep. So we are going to start with the second talk. Our next speaker is Lourdes. She is already here, and she's going to talk about exchange and expansion, a fish story. So about Lourdes Martínez-García is a third-year PhD student at the University of Oslo, working with the sustainable management of fisheries from a molecular perspective. Lourdes has been studying the dynamics of marine ecosystems for 7 years, and she had the opportunity to perform field work across her natal Mexico and Sweden. She has been an ocean lover since she was 4 years old, yet she shares this love with the rainforest. Outside of academia, Lourdes enjoys swimming, a good book, and chocolate. So let's hear her talk.
Thank you very much. Okay, first I just want to check that everybody can hear me okay. Yes? Okay. Well, hello everybody. Thank you for being here today. And also I would really like to say hi to all of our online speakers. I know there's some people online, so hi. So yes, you've heard about me, and my name is Lourdes. You can call me Lulu. So I'm a biologist. And that means that ever since I was little, I basically just love to look at animals and plants. But now I work with fish. And I spend most of my time working in front of the computer instead of actually being in the field. And well, that's what I'm going to talk about today. So I'm going to go and just dive into my talk. Okay, so just let me see if this works. Yes, it does. Okay, so exchange and expansion, a fish story. So I would like to ask you guys, which fish is this one? Anybody? Oh, yes. Okay. So this is Atlantic cod, and this is going to be the main character of my talk across like the whole night. So a lot of people say Atlantic cod is quite an ugly fish. In my personal opinion, I think it's quite beautiful. And yes, I would like to ask you, what do you think about when I say Atlantic cod? Anybody?
Fish and cheese.
Yes, so food, right? Well, a lot of people have told me before about books or even board games. This one is called Moonglow Bay, if you're just interested. I didn't know about them before I started working with this fish. So yes, a lot of people think about this, but most of the people actually do think about food. So there is a lot of different dishes that are based on cod. Quite delicious too. So if you think about different dishes made with cod, you might also wonder, how was this fish caught, right? So if you actually do wonder about that, you're then wondering about cod fisheries. So cod fisheries has a really long story, and especially here in Norway. And I can tell you that there is a sustainable way of looking at the cod fisheries, because if you know the when, the how, and the where these fisheries started, you can actually spot populations that were depleted in the past. So you can actually, like do something and make optimal sustainable measures to try and do better for the stock we're talking about. So if I go a little bit on a broad perspective about cod fisheries, I can tell you that around the 11th century, so here, all the way to the 12th century, around Europe, cod fisheries were mainly local, meaning that the people who would caught the fish, they would feed people in their local, in their coastal communities. And by the 13th, 14th century, they would, it started expanding across Europe, so more to the North Central Europe. And if I go more into detail in Norway, there was something really specific that made fisheries expand, and that was stockfish. So I'm sure a lot of you know what stockfish is, but it's this air-dry cod. And these fish or this product made out of cod allowed the fishermen to actually sail away with the fish and it would not spoil, right? So this was mainly produced in the Lofoten and the Vesterålen Islands. And this I'm talking about before, like in the Iron Ages. And we can tell you, as I said before, from the 11th century, it was mainly local, but everything actually changed by the 16th century. So how come? Well, the Americas came into the picture. So what happened is that from England, mainly Bristol, someone sailed away and ended up in Newfoundland. That's how they call it. So Newfoundland was this part of now Canada where they landed, and they actually said that in the historical sources, it's really nice to read how they describe that they would just drop a basket, lift it after a couple of seconds, and it would come up with like full of cod. So this place was just basically like full of cod. So here is where the history of fisheries and actually precisely cod fisheries changed completely. So what happened after this? Cod fisheries just went up. But what happened when something goes up? Well, sadly, it tends to go down. So what happened to cod? Well, it declined. Sadly, it really declined. So in this graph, I'm mainly just showing you like the dramatic decline it suffered in the 20th century. But it actually has suffered several declines across, like from the 11th century all the way until now. Some of these declines actually fall in between with either social events like the war, or it also falls with climate events like the Little Ice Age around the 16th century. So yes, cod decline, and some of the stocks across the world declined, for example in Canada, and until date some of them have just not been recovered. Others, like for example here in Norway, the North Southern cod is doing very well. Some are not. There you go. So when I talk about the sad history of the cod, people don't really think about the genetic consequences of these declines. So when I mean genetics, I mean DNA. So DNA can actually be about fisheries or fisheries annually, or can happen across the life of species. And I mean in the sense of like if someone is making your population smaller, then your genetic diversity is going down with you. So that information that is being lost might be crucial for you to survive, especially under a changing climate that we're of course living in. So this is exactly what I'm studying now during my PhD. So I'm looking at the demography, evolution, and also ecology of Atlantic cod through time and under a fisheries and climate change perspective. Yes, so to look at the past history of a species, it's not always straightforward. So we have to, of course, create methods that are useful for this, and that's how ancient DNA came into the picture. So maybe some of you are wondering, what's ancient DNA? Well, basically DNA, but just retrieved from a biological material. What's that? Well, it's just basically plants of animals, right? And this material can be, for example, zooarchaeological material. And what is that? Well, it's just basically bones, right? So these are some of my specimens. That's the closest I've been to a fish so far. So what we do is that archaeologists have all the fun they want in the field. They just go, dig, and get the bones, and they then send it to us. So we get the bones, we crush them, we get the powder, and we extract DNA. So there are many things you can find out through ancient DNA. And some of those things that I would really like to highlight are, for example, the changes that can happen across time in a population. Meaning that, across time, you can have a population in low level or in a high level. Maybe just in a high level or maybe just in a low level. And also, which habitat are they living at? Has it changed? Is climate change actually, like driving them in different habitats? So what's going on there? And another thing that ancient DNA allows us to do is to track historical trading sources. So why would we even want to know, like how historical trading routes look like? Well, as I mentioned before, if we actually know how they traded fish in the past, we can spot which populations they were taking the fish from, and then we can apply several sustainable measures to that specific population, right? So I'm going to talk about one of my projects in my PhD, where I am precisely or specifically looking at the origin, the biological origin, of samples of Atlantic cod retrieved from England and Spain. So basically what I did is we gather a lot of individuals of Atlantic cod, and we set out to find out where are they from, right? So the excavation was in London, specifically in England, and in Spain we have them across Madrid and Barcelona. So yes, the place where these fish can be, it can be wherever, right? Like as I told you before, it can be across the Eastern Atlantic. It can be either, I don't know, the North Sea, the Irish Sea, Celtic Sea, a lot of different places, or even the Western Atlantic. In the Western Atlantic, just remember, it's mainly before the 16th century. So that's exactly what we set out to do. And before I share with you my results, I would like to show you this figure where I would like you to focus just in the colors. That's the important thing, right? So the colors, as you can see, are quite different across locations. And why is that? Well, so Atlantic cod is a very well-studied species, right? So across time, people actually discover that there are 4 different regions in the genome that are quite different across like both the Western and the Eastern Atlantic. Those 4 regions are these 4 ones that are there. It's not important for you to remember the names, but there are 4 chromosomes, basically a big molecule of DNA. So these 4 regions, as they differ across different locations in the world, this means that, they're kind of related with temperature, salinity, migration. So we can really spot the fish because, for example, the Northeast Arctic, in comparison with the Lofoten coastal, they migrate differently, right? So the Lofoten coastal or the Norwegian coastal fish would basically spend all his life in the Norwegian coast, while the Northeast Arctic would go off to the Barents Sea, feed up there, and then come down to like spawn. So those are the differences that we can look into these specific regions. So thinking about those regions and the difference in color that we see in the squares, we found out our results. And those results are these ones. So again, here, just look at the colors. In the bottom part, we have the time. And here we have like the area where they are located. And in more detail, here we have Spain, which is, not Spain, England, which is basically London. So basically what we have here is that, as I told you before, at the beginning, we have mainly local fish caught, right? And then we start seeing a little bit more of diversity, a little bit more of color. So that means that fish from long distance sources were caught. And by the 16th century, we already see a little bit more of a Western Atlantic signature. So this is completely consistent with historical sources. But what we did find out is that there is a little bit of an earlier signature of Icelandic cod. So Icelandic fish was perhaps more important than we have thought, maybe. And then for Spain, we can see that our sample size is significantly smaller, but we can see that some of our individuals, if like the very first ones, are completely from the Western Atlantic. So these fish might have been caught by French or English people, which we know were trading the cod between Spain and the Western Atlantic. And then by the beginning of the early modern times, we see already Eastern Atlantic signature. Basically, we know Norway was already, again, trading with the Southern Europe. Okay, so basically what I would like to tell you from this talk is that there has been an increasing trade of cod, right? And that the demand, well, that what drove this expanding trade was the increasing demand of fish in different countries. And also that this exploitation was definitely local and from distant sources. So mainly the take-home message that I would like you to take is that the cod fisheries were actually bigger than we thought. And that is really important because sometimes we don't know how depleted as a population is now. And we actually, if we understand the past, we can actually perhaps make an impact in the future. So that would be it. And of course, I'm not working alone. I have a lot of collaborators, so I would like to thank them all. And also the ITM I'm part of, See Changes. And of course to you and to the people online. Thank you very much for listening. Are there any questions?
Sorry, sorry, I have some technical problems. Sorry, does anyone has questions?
How do you date the bones? How do you know how old the fish bones are?
There are two approaches. I haven't done it myself, but archaeologists, when they go out in the field in the excavation, because of the soil layer, they can actually know how old a fish is because of the soil, or infer in the context of the soil. And then the other more exact way of knowing the age is through carbon-14. Yes.
Thanks for the presentation. I was wondering, did commerce in a way affected genes between like the different areas, or how did this affected the different genes in different areas that you have code that is affected by?
Can you perhaps repeat the first part? Did what?
Yeah, how did commerce affected the different like genetic differences in all the different areas?
By commerce, you mean like fish, new fish coming into the population?
Yeah, trading. Thanks.
Aha.
or the communication. Either is it trading or is it that they take live fish to the areas? And so I was wondering how did this happen?
Okay, so there are two ways of answering that question. So it really depends if the fish was alive. So if the fish is dead, it really doesn't affect, or it could affect depending on how we're studying the fish. Because we might take, if we don't know where it's coming from, we could take its genetic information as from that position, right? So that is why it's really important to know where the fish is coming from. But if the fish was alive, then it could potentially influence the genetic information. Yes, I don't know how much, because I don't know how much alive fish we would be talking about. But if it reproduces, it would absolutely become part of the gene pool. So it would potentially affect.
Hi, everyone.
Hi.
Thanks for the very nice talk.
You're welcome.
I think I'm still hanging up a little bit on the how they aim to get the fish. But I'm also wondering, like, do you have very similar genetic information between types of fish? Because if they go down and they take something out, I mean, how do you know this is obviously?
Okay, so the way we know that what we're catching is cod is that we get the DNA out of it, and what we map means basically compare the genetic information of our sample to our reference genome. So basically a main sample of cod that we are sure it's cod. So what we compare is a mitochondrial DNA. Then I don't know if you guys are familiar with mitochondria, but it's basically this part of the DNA that is inherited from mom to son or daughter. So it's quite conservative to say, okay, this is cod. So if it matches with our sample, we're sure it's cod. That's the way we decide. Any other questions?
Yes. Hi, thank you for a interesting talk. I was wondering, looking at the highs and lows of the cod population, what large historical events do these highs and lows actually correlate with the last 500 years? Like, are there any sort of surprising events which makes the cod population really fall down?
So you mean the 500 last years?
Yes.
500 last years, I think it already includes the Little Ice Age, so when everything got super cold, and it might also involve the medieval warming age, when everything got super warm. So it actually does, definitely when it got super cold. I don't know if there was a decline when it got super warm. That I don't know, I would have to check it for you. But it basically overlapped with that one, and also with the war. Actually, with the war, it went up because nobody was fishing it. Yeah. So that's some of the examples when it goes up and down. Yes.
There's a question.
Yes.
Yes, thanks for the very nice talk. I have a question that actually fits to the last one, but looking not in the past, but in the future.
Yeah.
Like with climate change, the seawater temperature and chemistry might change, so it might change as well the roots of the cod. Do you expect as well, like decline because of climate change, and could you kind of use that method of yours as well for that?
Yes. Okay, that's a really interesting question because cod sometimes doesn't behave as we would expect. So that can be a little bit confusing. There has been some modeling made by other groups, other universities, and it basically shows how cod is just migrating north. It's not really decreasing so far in the modeling, it's just moving up north. But as we know, the further they move up, the faster they're going to run out of space. So at some point, my guess would be that they're going to start going down just because they don't have enough suitable habitat to continue living. But of course, it's still up to see what actually the results say about it.
Are they moving north because it's colder? Are they moving north because it's colder? Or?
Yes. Actually, yes and no. They're moving up north because the food is moving up north.
Okay.
So just related to the last one, so what do they feed on?
It depends. It depends which stage we're talking about. So if they're just born and juveniles, they basically would eat crustaceans and many other invertebrates. But when they're adults, they would feed mainly in fish. Cackling is their main or preferred source of food.
And then the other question, I would imagine most bones disintegrate over time. So how does it become a fossil, or why is it preserved?
Okay, so we actually don't work with fossils. We work with the actual bone. When it's a fossil, you have it more of a, how can I explain it? I'm not a fossil expert, so there you go. But it doesn't look. Oh, I don't have that picture. It doesn't look like. No, it's maybe. It won't look like that. You have it more imprinted over a surface. That's a fossil. but these are just the bones, and it depends where they were retrieved from, because sometimes the soil with what would be the word? It kind of consumes them, like the pH, for example, as someone was like, it was mentioned in the talk before, it changes, so it could actually affect them. So it depends where we're getting them, the quality of our bones. Any other question? Okay, the oldest bone, I think, is around more than 4,000 years ago, like 5,000 years ago. That's the age.
Do you think it's possible to start from older bones?
Yes, I know. I don't remember the exact age, but I know there was a paper published about mammoths, and I think it was like 1,000,000. I think you would have to check because I don't remember, but it was way more, like way older than that. Yeah.
Is it easier? It is more difficult to extract DNA from the older bones.
Yes.
Then how do you assure that it's? It's just a lottery, right? In the end, that you take the bone, you smash it, you extract it, and then it's like.
Oh yeah, you mean the success rate?
Yeah.
Yeah. It definitely depends a lot in what I was saying, like how conserved the bone is, like how many contaminants it have. So yeah, you can get 10 samples, and from those 10, 3 are going to be useful, maybe, or maybe the 10. You never know. Any other questions?
Yeah. No, but then when you think about it, the bone is solid, right? So in theory, when you crush it, the DNA should be, when I think about it, it should be very intact-ish. I mean, I understand corrosion, etc. But like you have these bones, and I would think that if I crush it, then DNA should be there, right? So
Yes.
why it's more difficult? I mean, I don't know if you know the answer. Why it's more difficult to extract the DNA from older samples?
Because of the contaminants. Just because if it's quite for example, if you would extract DNA from me, it's quite easy because I'm alive and nothing has degraded, right? But these guys have been dead for quite a while, so the DNA has degraded. So the opportunity for us to actually retrieve that depends a lot in how that was preserved. So if it's very well preserved, we would have no problem in getting it, or maybe not so much trouble. But if it's like, I know that there are some samples that have been exposed to like flops, for example, it's really hard to get DNA from those. Okay. If there are no more questions, I'm gonna just pass on the microphone. Thank you.
Thank you very much, Lourdes, for your talk. So we are having another break. So 10 minutes, we just go and grab more beer. I go to the toilet and, well, just come back, please. Oh, yes. Are you ready for the last talk?
Yeah.
Well, woohoo!
Finally, last one. How long?
So our last speaker is Agnieszka, and her talk is what happens to soap after wash your hands. I will read something. I just want to clarify that I didn't write this.
Yeah, it was me.
Agnieszka started her scientific journey in Poland through Spain, French part of Canada, and ended up in Norway. She began as pharmaceutical biotechnologist, but she switched to environmental one to fight the emerging pollution. During her PhD, she was trying to remove pharmaceuticals in water. Now she is a Marie Curie postdoctoral fellow at NMBU, where she is developing a monitoring system for pollutants that are coming from our daily lives. She is a crazy penguin lady, as you can see, who in her free time is traveling around. So let's hear the talk.
So first of all, thank you all for coming and staying till now, because I'm the last one, so it's the worst, I guess. So last man standing, congrats. So yes, I would like to talk about what happens to soap after we wash our hands. So I guess all of you use soap, right? Shampoo, body lotion, etc. But who thought about this question before you saw it, of course? Really? Did you come up with any conclusion? Okay. So basically, like the easiest answer to it is, oh damn it. Come on. Oh, okay, it's coming. Yeah, so basically it's going with the pipelines. So wash your hands, taking the shower, I mean the shampoo and etc., shower gel. It's, oh my God. It's going with the pipelines to the water treatment plant. Where of course the water is treated. So all the things, because if you can imagine, if you take your shampoo or soap or whatever, you turn it around and then you have ingredients. So they're like, I don't know, 5, 10, 20 ingredients. So of course it's a foam and everything, but then everything is going to the water treatment plant. So yeah, it's going with the pipelines. And imagine that this dark blue, that's the main pollution. So I don't know if anyone have ever visited wastewater treatment plant. Yeah. So it's like a sludge or the mud. It's stinky, that's the other part of it. But imagine like this muddy sludge that is going down the pipes. So that's the representation of the dark blue. The small dots, that are the ingredients of our soap or shampoo, body lotion. So what is actually happening later, that during the treatment in the wastewater treatment plant, so before the treatment we have a lot of goo and sludge and mud with a small amount of these ingredients of the soap. But after the treatment, what is happening, like the the dark blue, it's decreased. But the pollutants, the ingredients of soap, they actually, you can see it's, I mean, yeah, here it's like 4 versus 4, but usually it's like, I don't know, 5 versus 4. So the concentration, the amount of these ingredients, they do not decrease in such a manner that we would like to. Why? Well, if you can imagine a dodgeball, okay? So the main pollution, a lot of it. So it's easy to catch it during the treatment. Whatever it is, if it's like mechanical filters, whatsoever, it's really easy to see it and you can easily hit it. Well, if we are talking about micropollutants, so the small dots that we have seen, that's the case. Okay, sometimes you will hit it, sometimes you will not hit it. Well, basically it's probability and statistical analysis. Yes. And then what's happened actually? Well, come on, go back. So, okay, main goo is gone, the main sludge is gone, the micropollutants, they are staying. So what is happening? Well, basically, after treatment, everything is released to the environment. So in case of Oslo, Oslo Fjorden. In case of different cities, when I was living in Quebec, it was the river, but lakes. So all these pollutants, of course, they are entering here. And we are talking about big pollutants, so nitrogen that we are talking about today, or phosphorus that is also used, it's important, but it's a problem if we have it too much. Well, we have regulations for them. It has to be below a certain level. And this wastewater treatment plant, they have to do it, and they do it. If we are talking about the small things, well, we have right now regulation for some pesticides, one plasticizer in drinking water, and another 20 compounds. That is a group of compounds that are used for the flame retarding things, but this is one group of compounds, so not so much. So, okay, I was talking about, it's a small concentration, but what a big concentration for the sludge and main pollution. But what is actually this small concentration? Because everything is relative, right? So I have a question for you, if I can make it. So when we are talking about water pollution, we have 4, let's say, main units. So we have grams per liter. So if you can imagine a bottle of beer and like 1 drop of water, that's gram per liter. Milligrams per liter, so 1 drop of water in 2 bathtubs. Microgram per liter, so 1 drop in the swimming pool. Or nanogram per liter, so 1 drop in a lake. So what do you think, what is the concentration of the pollution that are coming from soap? Like in the environment, like when it's released, it's not treated, right? But honestly speaking, the concentration of emerging contaminants, of the pollutants from daily life, the range in influent, in sludge, and in effluent, it's more or less the same. In case of this. Milli? Okay, so who thinks it's milli? Micro? Nano? Yeah, so basically micro and nano. So that's the pollution. If we are talking about this main pollution, big pollution, that's grams and milligrams, actually. So it's crazily small if you think about it. But so actually, why should we care? Like, that's the basically, yes. I started my talk about soap because this is what we use. All of us we are using, but this group of contaminants that are called contaminants of emerging concern, because well, they belong to these kind of categories. So we have personal care products, soap, pesticides, so fertilizer, etc. Plasticizers. So if you have the plastic bottle, you can see. I don't know if you can see actually, but especially in North America, when you have plastic bottle and you buy it, it's like BPA free. So BPA, it's one of the plasticizers. It's super toxic for us also, and it's banned. That's actually one of the emerging contaminants that is included in European Union directive for the drinking water since 2019. Yay! So yeah, endocrine disruptors is the plasticizers I was talking also about. Antibiotics, so all the pharmaceuticals, biocides, textiles, pesticides. Yeah. So it's like I said. Why should we care about it? It's such a small concentration. 1 drop per lake. Come on. Well, the thing is that, yes, we have started to see it. It's an increasing trend because, as you can imagine, there is a globalization, so we need more meat that we have found out on Monday in one of the talks. We produce more, we need more. So, of course, the pharmaceuticals, for instance, in this case, we are using also more. So the more we use, the more we are discharging to water. Well, the problem with pharmaceuticals, the biggest one, are antibiotics. Because as you know, you take antibiotics when you have bacterial infection, right? And you kill bacteria. Fine. Well, then imagine that the antibiotics, they are going to the water and they are at this low concentration. And then we have bacteria in soil, in water, also in lakes, river, etc. And the concentration, this micrograms, nanograms per liter, they won't kill all bacteria that are in river or in soil. So what doesn't kill you makes you stronger. So here we have antibiotic resistance. And to show you why it is a problem, oh wow, about pharmaceuticals also. I'm sorry because I did this. Oh, never mind. So about the pharmaceuticals and antibiotics, what is also important, I mean, and it's actually a shocker, when you take any pharmaceutical, so antibiotic, ibuprofen, paracetamol, whatever, well, part of it is absolutely absorbed by your body. So like, let's say 50%. But then another 50%, when you go to pee, it's going unchanged. So imagine you take one pill and half of the pill is, yes, of course, it's in your bloodstream, but the other half, you are just peeing out, and then it's going to the toilet, and then pipelines, and then water treatment plant, and then we don't treat it 100% because treatment plants, they are not designed to reduce such small amount of pollutants. Also, the other problem with pharmaceuticals, well, many people, they don't know how to dispose it. So we are just putting it into the drain because it's expired, or we put it in the dustbin, which is not good also. And then everything in the end is going to the water. To show you the problem, finally, maybe, or not. Yeah. So there was this calculation that they did in 2014, actually. There's a simulation that says that antibiotic resistance, antimicrobial resistance, that will be the main cause of death in 2050. Yes, in 2050. So it will beat cancer. To also tell you that it is really the problem about the pharmaceuticals, and pharmaceuticals are my favorite topic in this case because I did my PhD on it, and it's really depressing. So, yeah. In a minute. So, yeah. So basically, for instance, because yes, we are using antibiotics to treat our diseases, but then there is this big thing that agriculture is using antibiotics, of course, at low dosage, to boost the meat production. Okay, so if you feed chicken with antibiotic, it will grow, I don't know, 50%, 150% faster. As I said, everything is increasing. So pharmaceuticals increase usage in Germany of this much. What I said before, 30 to 90% of the pharmaceuticals, they are extracted. So we are peeing it actually and pooping. Oh, yeah. So that's pharmaceuticals. Okay. I said also about hormones or the compounds that they are disrupting our hormonal systems. So low concentrations, okay, long concentrations of the hormones or BPA, bisphenol A, or estradiol, so the things that are coming from the contraceptives pills. Well, they were detected also in lakes and rivers, etc. Low concentrations, okay, and people, they noticed that there's a feminization of fish. So suddenly there was a change. They have seen the changes with the fish sex, which is actually not good. Also because I'm not the expert on fish, but I suppose that they cannot reproduce, and that's the main problem also. Yeah. If that doesn't concern you, well, then maybe this slide will, because the small concentration of all the emerging contaminants, they can influence us. So this is the effect on human health of PFAS, so perfluorinated alkali substances, that are also regulated from 2019 by European Union. So what can they do? So basically, yes, development affects unborn child. We can have breast cancer, liver damage, kidney cancer, testicular cancer, increased time to pregnancy, well, basically this obesity, and that's just one group of compounds. And reminder, low concentration in water. So basically, when I found out about it, yeah, that's how I felt. And let me tell you, I found out about it by the end of my master's studies. And my master's studies was pharmaceutical biotechnology, when I spent 6 years to learn how to produce pharmaceuticals. And then I realized that, okay, I'm learning how to produce it, but then I'm releasing it to the environment. So then in the end, I'm producing more disease. So it's like, it's like a vicious cycle. So I was like, no, I will not contribute to that. It's better to prevent than treat. So basically, this is what come on. It's so funny with this, So this is what I do. I am biotechnologist, like I said, pharmaceuticals in the beginning. So biotechnology, for those who don't know, using whatever we have in our world, so bio, like for instance, the group with the nitrogen, to change it to technology. So like I said, I switched to environmental, yeah, I think so. Thank you. To the environmental biotechnology. And during my PhD, I was actually testing different kinds of materials to remove the pollutants from water, or to see how it is affecting the real life. I mean, the aquatic life in that case, because, well, it's easier. Yes. What I'm doing right now, what I'm doing right now, and I did before also, it's like, how do we know? I said it is like so long, so small concentration. So how can we see it? Well, it's a long process. Does anybody do analytical chemistry? Yeah. Yeah, so basically, you target your compound. Okay, so it's like pharmaceutical A, let's say. Then, for instance, you have to take 1 liter, because you have to be sure that you have it in your sample. Then you have to concentrate it. So you go from 1 liter to 1 milliliter. Then you go with the fancy machines that are super expensive. Not everybody has it. You have to be really trained to do it, etc. Then you go to your computer, you take the results, you have to, of course, recalculate them, and then voila, you have the results. If you're lucky, you can do it in 2 days. If you're unlucky, because, for instance, I don't know, you have to wait for the results, etc., etc. I don't know, the worst, I was waiting a month. So that's the reality. So what I'm doing right now, yes, thank you. Yeah, it takes time, yes. Yes. Yeah, so what I'm doing right now, and I'm trying to do right now, is that I target certain pollutants, and I try to find the butterfly effect. It doesn't mean that I'm going back to the past. It means that I'm looking in the water treatment plants. I'm looking for the chain effect. So if, let's say, contaminant is going to the treatment plant, it is affecting the sensors like pH sensors, temperature sensor, oxygen concentration, and I'm looking for the domino effect, or the butterfly effect. So one sensor is triggered by the change of one sensor, it's triggering the change of a different sensor, and like a domino. And then the idea that we wanted to have, it's like still in the beginning of the process, so I cannot answer you. I cannot answer many questions about that. But the idea is to create a software sensor. And you can, yes, so that ideally we can sit at home and just watch how is the concentration of our daily pollution. Yes. So this is what I do, but what you can do, because that's the important question also, because we cannot do it alone. So first of all, Well, baby steps. Okay, because when no, not yet. I haven't finished the thought. So because when I heard about it, I was like, damn it. I'm studying to produce drugs. That's the first thing. The second thing is that with my daily life, I'm contributing. I'm washing hands and I'm contributing. I take shower, I'm contributing. And like, okay, from now on, I'm taking a quit life. I'm going to the forest. I'm going to build the hut with my own hands, and I'm going to live like a life that my ancestors from, I don't know, thousands of years ago lived. Well, basically, that is not a solution because I will not fix anything. So it's easier to reach it by baby steps. And these baby steps are, yes, first of all, dispose medications properly. So if you have expired meds or you don't want to use it anymore because anything can happen, pharmacies, even if you don't know, because I know every country has it differently. I know in Spain they have like big promotion of it, that they are big bins that they put it there. But if you go to the pharmacy, they will tell you what to do. And actually, probably they will take it because they have a special measure to deal with it. So that's the first thing. Yes, next one. Well, reduce the chemical use in your home. Never mind. Because even the other day, I was reading about article about washing your hair. And it is said that you don't need thousands of liters of the shampoo in your hand so that you have the beautiful foam. You just need a little bit because it's better for the environment, but the second thing is better for your hair also. And you don't need like crazy amount of soap to wash your hands also. It's just like a one chink. It's enough. Next. Yeah, be careful how many things you use. I'm pretty sure that if you go to your bathroom, let's say, well, you have, let's say, soap for hands, soap for feet, soap for body, and I don't know for what else. So, soap for the weekend, soap for the everyday. Well, if you take it, the composition is probably the same, or you can actually use the same thing for washing everything, more or less. So you don't have to buy, because also it's a little bit of greenwashing. No, it's not greenwashing. It's the marketing thing that companies, they're like, okay, we have this beautiful product that it's, if you use it and you wash your, I don't know, feet, it will be so soft and everything. Well, it doesn't make a difference probably because it's just the marketing thing and the composition is the same as any other soap. So yeah, that's the thing to remember. Next. Yeah, be careful how many things you have inside. So less composition, the better. If you think about it, less composition, the simpler the better. Less composition, less is going to the environment. Additional, if that doesn't say anything to you and doesn't, you don't feel convinced about it, then think about it. That all the like there's a trend right now that we are using so many chemicals that we have many allergic reactions to it. So yes, environment. Wait a minute. So environmentally, this is a better idea, and for your health, this is a better idea also because less allergic reactions. So two things. Next. Yeah, be careful. That's greenwashing. Be careful what you buy. So right now you go to normal or whatever, and then we have 98% of natural things, and our I don't know, antiperspirant deodorant is super eco, and everything is green. Well, that's bullshit. Not with everything, I'm not saying, but before this green trend happened, let's say 10 years ago, you're using the same Nivea soap, okay? And right now, your Nivea soap is like 98% of the organic compounds or 96% of organic compounds. Well, the composition hasn't changed. They are just informing you that, yeah, the composition that they are using, the ingredients, they came from nature 10 years ago, and they are still organic. I mean, they're still, Nothing has changed. They're just informing you to buy it because you think, okay, it's organic or it's bio, so it's fine. So yeah, be careful what you are buying. And in the beginning, I was so ambitious that when I was going to the shop, grocery, and I was buying things, I wanted to Google all the ingredients to see, okay, if it's toxic or not. Yeah, seriously. So you don't have to do it. You have applications. So, for instance, INCI Beauty, this is a French one actually. This is the EWG Healthy Living app. Cosmetics, that was they did it during the European Union program. But also you can look for the logos. So for instance, Eco Logo certification, EU Eco Label, and here you have, it's like there are certifications that really, that means that first of all, the composition, it's more environmental healthy, but also the production, because that's the other thing that we don't think about and it's important. Yes. And I think that's the last one. So the popular one, reduce, reuse, recycle. So reduce, of course, in general, but in case of chemicals, reduce the usage of chemicals. Reuse, well, that's actually difficult, but if you think about plastic bottles that you can use or reuse, that's a good thing for the environment also, because microplastics, they are the problem also. And recycle, well, that's pretty clear. And that's it. Yes, you can click. Yes, thank you so much.
So thank you, Agnieszka, for your really nice talk. Is there any question from the audience?
What soap?
Actually, I have 4 questions.
Oh, damn it.
Do they take into account metabolites in the nutrition?
There's
Now there's a new trend to use natural products. So do they take that into account in the detection or the effect of natural content on the human body due to the complex chemistry? The third is what is the delta time between the molecule and the implementation legislation? And fourth, what about the are they coming from science or are they real science? Just
this is the thing.
Just want to try something and it was that good and we do it.
Yeah. Oh my God. Yeah, I will. Okay. I will start from the last. So the applications, because people ask, okay, is it like a random application that anybody can do and it's not based on science? Answer is the applications that I said, they are based scientific. It's the this one, for instance. Come on. This one is it was the European project, and it's science based. They updated. If it's like you take the phone and you scan it, you scan either the ingredients or the not QR code, the barcode, and then of course there are not all of the cosmetics are there, but then they you can upload it. That okay, we don't have it in our database, so take a photo and we will check it for you. Of course, it takes days or something because, well, it's not automatic. It's not automation. But yes, people they are the uploaded cosmetics. People they are checking it based on science. Natural emerging contaminants. Yes, that's a separate thing also. Because yes, the main the things that are coming from our daily life and what we are producing, the synthetic things, it's the one thing that is toxic. But the natural things, they are toxic also, and it's taken under consideration. So it's not that only the man-made things are ecotoxic for us. They are taking under consideration also the natural ones. Yes, yeah, and that's the other thing about be careful what you're looking for, like 95% of organic things. Yeah, but this is taken under consideration too. Metabolites, if it's taken under consideration also, that's a big problem. Okay, that is like a humongous problem, and this is like this is the problem also with monitoring, because If you take the glass of water, even the water that you have right now or from the tap, probably you have the cocktail of everything. So there is the huge scientific analytical chemistry part that has super fancy machine that I wanted to send my samples to, but we cannot do it because it's too expensive and it takes so much time. I don't even have, I have the beautiful graph that shows it. There is European Union says that we should take in the cocktail effect under consideration, and whatever we can do. One thing to do it is to do the biological tests, so that you take the water, you take the sample, you introduce it to bacteria or daphnias or plants, and then you have the cocktail effect. But in the end, you don't know what is inside. So that's a huge problem, and that's a different department also. And what was the first one? Between the of the you are talking about that I discovered something today. Like I discovered today the pharmaceutical and
New molecule and then legislation on it.
it's like that. Today I discovered cure for the cancer. I think that my molecule it will be the cancer. So then it's 15, 20 years of research.
No, but
For this one.
For the environment.
Okay, so that's sad, and I can tell you that. There's like a big list of what is inside in every country, even in Norway, like Oslo Fjord. You have pharmaceuticals at low levels, and yes, of course, there's a lot of research, but simply it is not affordable to regulate it right now. There is a monitoring system, so they upload all the information about it. Research is like a big database, and they say that, okay, we discovered this much of pharmaceuticals, this much of the plasticizers, this much of this thing. But because it's a low concentration and it's not killing it right now, like you drink water and it doesn't kill you. So that's the problem for another day. Because if you have to, there are amazing technologies to treat water, but they are expensive. So water treatment plants, they would have to upgrade to remove it. If they have to upgrade, then have to put money, and they don't have money. So it's changing. It's changing. It's like there's more pressure put on it. So like I said, three emerging contaminants, they came to the directive, drinking water directive in 2019, and they are watch lists also. So it's monitored, but because it's not killing us now, it's just FYI. And let's keep tap on it. I know.
Well, I have one question about, you mentioned microplastics.
Yeah.
So how is the situation with that? Is there actually technology being developed to try to remove them, or we should? Yeah. What is your opinion about this?
It's not an opinion. The research is happening. So how to detect it, how to remove it, how to classify it. But microplastics, it's such a big thing that there are no legislation about it, because they don't know, it's so diverse that they don't even know how to characterize it. So it's like people, they are saying, okay, we discovered the compounds that are polypropylene, polyethylene, etc., or the mixture, but they are just, this is classification that they use. There's no even proper classification of it. So there are studies that are treating it. Of course, it's like the research base. I didn't deal with there are the microbiological, for instance, the bacteria that eat plastic. But legislation, as for now, there is none. The only thing is that reduce the use of plastic. Yeah.
Yes. thank you for the very nice talk and the inspiration in the end of the meeting. So I was wondering, this polluted water, then it goes into the river and finally in the ocean. Can you still measure like concentrations of like all these pollutants even in the oceans, or like even like at the poles, or like what's the research state there?
Yeah, easily. You just have to take the big enough sample. I'm not doing the ocean part, but yes, you can just. There are people that are grabbing the sample, and they are measuring it easily. Because the thing is that like all the waters are connected, right? So sink, pipelines, water treatment plant, river to sea, sea to ocean. So if the compound is like this perfluorinated alkali compounds, they are like forever chemicals. It's really difficult to degrade, so they can easily like travel.
Thank you for an interesting talk. I have two questions.
Yeah.
One is about the labeling. What do you think of the swan label?
Huh?
The swan label.
The one Norwegian one.
Because in my naivety, I thought that was good. But maybe.
No, I think I was checking it. I also like follow it. There's also like there's the website. I don't remember it right now. I follow it. I don't remember why exactly. I'm sorry, but I was checking. I was cross-checking it. It's because it's for the Nordic countries. So yes, me personally, I go with it. I seriously don't remember why now. I know that there's a website that says global labeling, and it's like official website, and there are all the labels from all over the world, and they are like certified labels that you can use. I think I was checking this day. That's why, because some things I check and I tick and I don't remember why.
And the other one is, are there any rings for this in, not in the wastewater, but in the other end, like for drinking water? Do they have rings for it in plantations, or when they rinse drinking water? Because you say we get it in the glass when we drink tap water.
Yeah, we do. It's the same thing for the wastewater, for the drinking water. So, for instance, you have the source of the drinking water is lake. So, in Quebec, when I was doing my PhD, so you go, there's this point A in the river, and that's a wastewater treatment plant. So, treating, and then release effluent is to the river. And then downstream, I mean, if you go, there's a drinking water treatment plant, and source is from the same river. Of course, treatment is different. The restrictions for the drinking water are strict. I mean, it's stricter in the drinking water, of course. But because there are no regulation for the emerging contaminants, you have it. They are reduced. I'm not saying that the concentration is high and it's not removed at all. It's like really depends on the contaminant. 50%, 30%, 90%, but you have it. There's no, you cannot even rinse it, I think. Okay, I guess that's it. So, yeah, thank you so much and have a nice evening or the rest of the week.
So I think that now we are all impressed, but I hope that you have enjoyed the concert today. And well, this is our last night. We hope that you can come next year, but I want to tell you that we actually have more events during the year. So if you want to join, just check in.
Yeah, go on.
We have
Oh, I don't have it. I have it this one.
Yes.